All right, so here are supplemental notes for 2.2. So that supplement your reading notes, your textbook, and what we've done in class. And there's a couple other podcasts already up about these ideas. So what you should know is the prokaryotes are smaller than eukaryotes, and they came first in terms of evolution. Think about the endosymbiotic theory as we've talked about in class. Pro means before, karyote means technically like kernel, which we refer to the nucleus as. So there's prokaryotes and eukaryotes. EU means true. So prokaryotes came before the nucleus, and that fits in very nicely with our explanation of the endosymbiotic theory. So that being said, there's a tutorial that's in this document that's up on Blackboard that you can play. You can go through and look at the structural differences of prokaryotes, animals, plants, and building a cell if you want. But this will be a good review because you are responsible for being able to draw, label, and annotate diagrams of cell structure. So you should practice that. So that's a good first attempt. So this is an ultrastructure of E. coli. And what you need to be able to do is draw that and label, and as well as annotate. So here's a drawing with labels. Notice that the lines are straight, which is key, you must do. And you need to be able to draw this. So you have to include a flagellum, a nucleoid, cytoplasm. Notice that the nucleoid kind of looks like a nucleus, so I wouldn't draw it like that. I draw it like thread and label that as nucleoid, circular DNA. You gotta have a plasma membrane, a cell wall, pili, and ribosomes. And that would complete your drawing of a prokaryotic cell. But you need to also be able to annotate the functions. And what's important is that here's a little note, these cell parts are not membrane bound like the Golgi apparatus or endoplasmic reticulum that is common in eukaryotes. So they're not technically called organelles. Organelles must be membrane bound. So there are several functions that have to be able to be carried out. And here are the functions. You should be able to annotate this drawing with these functions. And let's see if we can get them up here. So this is what matches up. So you can quiz yourself, you can test yourself on this over and over and over again until you get 100% on this, okay? And then you also have to be able to, if you're given a diagram, be able to identify the structures. And as you can tell, this drawing is far easier to identify the structures than the actual micrograph. But you should be able to do this with both of them. So you should be able to, again, knowing how to draw these things, this should be easy. You know where the pili are, the nucleoid, the ribosomes, the plasma membrane, the cell wall, and the flagellum. And notice that this one has more than one flagellum. And then you need to be able, it says to state that prokaryotes reproduce by binary fission, but you might as well know what it is, and it's fairly simple. It's much easier, much simpler than mitosis, which we're going to learn about soon. But here the bacteria has a DNA loop, the circular DNA, the nucleoid. Well, that nucleoid goes through DNA replication semi-conservatively, very similarly to the DNA replication that we've talked about before. And then those divides are pulled apart to separate sides of the cell through proteins, essentially tubes, that pull those circular DNA loops apart, and then the cell divides right down the middle, thereby making two separate bacterial cells that are identical to each other. Now, I'm going to go back just a quick second to the functions here. So if you notice that the pili is for DNA transfer, which actually is very interesting because so bacteria can exchange DNA, they can share DNA from one cell to another. So if one bacterial cell doesn't have a gene, let's say for an immunity to some antibiotic, another bacteria can give it a copy of its gene so that it will be immune. And we see that happen all the time. So that's actually a little wrinkle into our natural selection explanation of evolution, which we'll come to. Just foreshadowing, if you want to look that up, look it up. Maybe, in fact, kind of Lamarckian, if you remember Lamarck, although Lamarck didn't know any of this. It just kind of fits with what his explanation was in some ways. So, good thing to know is that when populations grow by binary fission, they grow exponentially. Here's a diagram. Remember what an exponent is? Like x squared. So that it grows, the rate increases and increases faster and faster and faster over time. At least it grows exponentially until there's a limit on the resources like space or food. And so here is what we typically call like an S-curve. And that S-curve really stops right here, somewhere along the stationary phase, is what we typically draw. So this is called a J-curve, and this is an S-curve. So we start with a lag phase. There are only a few bacteria. Once they start to divide, there's extremely fast growth or increased rate of growth. So it gets faster and faster and faster. And then, once there's a limit on resources, the population can't increase anymore. So as soon as some die, there are others to replace them. And so the population size does not change. It just stays the same at a stationary phase. And that's typically where we stop with our diagram, and we'll talk. That's where we're going to stop when we get to our evolution unit. But there's a death phase as well. Once all the food is run out, and no more food, if no more food is being made, there's a death phase where the population decreases, actually quite rapidly. And then here's some gram staining. I'm not going to go into this. This is more for HL kind of thing. We're going to skip this, but you're welcome to read this and use any of these resources if you want to know more about gram staining, which is the way to identify what kind of bacteria we're identifying, or what we have. Here's some electron micrographs. That's what EM is, electron micrographs. So not things that we can see with the light compound microscopes that we have in class, but images that you can see through an electron microscope. And so there's some links here. And we're going to end with a little joke. A bacterium walked into a restaurant. "I'd like a pizza, please," he said. "I'm sorry, we're not doing food today," the waiter replied. "Why not?" "We don't have the staff." And we're going to end right there.